Good day, everybody. My name is Vasanth. I'm a NEET faculty who teach NEET aspirants and who train them for NEET aspirants, I can say. And today we're going to continue the next topic on how do organisms reproduce. So when we actually go into this topic, the previous topic, we studied something about asexual reproduction, and we have some parts of it, and we are going to continue with the same. And after that, we are going to learn a few more topics. So let's get on to it. Okay, shall we? Now I hope everybody could see. Okay, the topics that we are going to cover now would be asexual reproduction, a part of it, okay, which includes vegetative propagation, which includes artificial and natural ones, and tissue culture, and sexual reproduction introduction, and sexual reproduction in plants, and male reproductive units, and then female reproductive units. And after that comes pollination. Okay, we are going to study all these things in detail. So let's get on to the first one. So vegetative propagation is including two types. One is natural and artificial. So when you talk about natural, so natural includes the buds, okay, auxiliary buds that grows in the lateral shoots and develops into own roots and known as adventitious roots. So when you talk about vegetative propagation, so there are a lot of plants which actually grows on their vegetative part. So what do you mean by vegetative part? So when you talk about vegetative propagation, let's divide them. It's already two words. Vegetative represents the vegetative part of the plant, which includes the root, shoot, and the leaf. And propagation means growth. So growth that is enabled in any three of these parts, which is vegetative propagation. So the plants actually grows from the seeds. So seeds are formed from the flowers, and they are enclosed in the fruits. So that's how the process actually goes. So when you talk about the vegetative propagation part, there are two types. One is natural, another is artificial. So when you talk about natural, it involves several types of propagation, which includes bulbs, rhizomes, stolon, and tubers. So when you talk about root, so root includes sweet potato and guava. So these are two examples which comes under natural. So under natural, there are three. One is root, stem, and leaves. So these parts are the vegetative parts, which is going to give rise to new individuals or new plants. So when you talk about roots, the sweet potato is a good example from which a new plant could be formed, okay, if it is cut off. And guava, the guava roots, it can be planted and it can give rise to new plants. That's an example. And when it comes to stem, there are two types. One is runner and stolon. That's the primary differentiation. So runners and stolon. So stolon example is strawberry. So stolon is a part where there will be the main stem that grows under the ground, and from it branches arise vertically. Okay, from that vertical branches, we get flowers, and from that flower grows out to be and develops to be the fruit, strawberry. Okay, so this part is called stolon. And after that comes runner. So runner is where example would be grass. So how does it happen? It gets planted. So let's say we have a grass plant over here, and this grass plant will have a shoot that goes above the ground, and again it gets contact with the soil again, it develops roots, and then again another stem reaches another place. So that's how it actually happens. Okay, so that is a runner and the stolon. So when you have underground stem, example would be potato and onion. So when you have underground stem, the potato is a very good example. How? Because potato is just modification of stem. So it might be shocking for some people, but it is true. It is not a root modification. It's a stem modification. So how does it happen? The stem of the potato plant, okay, that actually gets the scientific name for potato is Solanum tuberosum. So this Solanum tuberosum, the potato, that gets submerged, okay, under the soil. So when they do so, the requirement of photosynthetic pigment chlorophyll will not be required. So that will be lost, and starch molecules that is produced by the plants is stored in the potato. So that's what we harvest and we enjoy. And then goes the onion. So onion is another example. So when you talk about onion, so onion has some structure like this, okay, at the bottom, and then a bulb-like structure like this, okay. So this part is the stem, and this part, the circular disc-like structure, is the root. That's it. That's the root. So if you just cut the onions like this, and then if you plant them over here, so the roots, new roots will be developed from the nutrient from the stem, and then this onion will grow back to the same. Okay, so that's how the structure will be like, very helpful. So next one is leaves. So how is leaves could be like part of vegetative propagation? Bryophyllum is a very good example. I have one in my lawn. Okay, so this bryophyllum will develop buds. So what do you mean by buds? A newly formed smaller plantlets. So where are they formed? They are actually formed in the leaf margins. So you can see a beautiful examples over here. So when they develop to become an individual, a self-sustaining individual, it drops from the margin and then grows out on to be a new plant in the same place. And this is a very good example. This was introduced to me by my mother. Okay. So now when you talk about a potato, how do potato actually go through? So this potato actually have some structures that looks like an eye, that is called eye of the potato. So this actually has a germinating tissue. So the nutritive material that is found on the potato will be used up, and this germinating eye bud will be forming the new shoot, and that will develop into a plant. So you might be thinking, what is this? Because this is just a stem modified into the potato, right? So that is nothing but in meristematic tissue you study three. One is apical meristem, lateral meristem, and intercalary meristem. Exactly. So it will be containing that particular structure. So that nodes will be forming eyes, and these eyes will be developing into a new plant when it is planted as such. Okay. Yeah. So this is the vegetative propagation when it comes to natural part. So there are artificial parts where it includes many steps. So where it involves human action. So the previous things which we saw in the natural doesn't require any human action. It will happen all by itself. But in the case of artificial, there are some more meaningful or necessary actions that we can do that can develop all these things. So it involves several things such as cutting, layering, grafting, suckering, tissue culturing. So all these things we'll see one by one. Okay. So the first one is the artificial is cutting. So cutting is where a plant is detached from the parent plant. Okay, a plant part. Okay, a vegetative plant part is detached from the parent plant and is planted elsewhere. It grows out to be a new plant. So when you get a rose, stemmed rose, after some time, after you enjoy the rose, okay, you just get the rose after it's dried off, take it out, cut it out, and then plant it in a nutritive medium, especially a potato with honey in it. So that you would have seen in several videos and as DIY projects. So that will enhance, nourish the stem to grow out, and you will get a new rose plant just from the stalk or the stalk of the rose. That become useless usually after the rose goes dry. Okay. So next one is root. From the root part is dahlia, that is actually a plant. So that plant produces beautiful flowers and stuff. So when that is planted, the root is taken out and it is planted elsewhere. The root will start to produce. It will have some vegetative propagation from the roots. So example would be dahlia. Okay. Next one is layering. Layering is a process where from a pre-existing plant, okay, we bend one of its branch. Okay, before you bend it, you strip the outer bark, only the outer bark of it, and then make sure it contacts to the soil. Okay, and then we fix it with such a support. So when such a thing is allowed to happen for weeks and months together, the plant bark, where the bark is stripped, so it will start to develop roots. So these roots will actually be supporting for the growth of the material. So this process of bending the branches towards the soil is called mound layering. So when you take the nutritive medium, the soil or the nutritive medium, to the air, so that you bound it to the stem by following the same procedure of ripping the bark of this place and then tying it with the nutritive medium, then is called air layering, because we are taking the medium to the place. So after a few weeks of development of roots, we can cut the shoot over here and we can plant them into a new plant completely. So that will be very good one. And after that comes grafting. So grafting is a method where we will be able to combine more than one plant. Okay. Sometimes it could be same variety, sometimes it could be a different variety from one another. So in that case, it involves two structures. One is the plant from which it is taken. Okay, the plant from which it is taken, and the plant which the taken plant is going to be implemented with. So the plant from which it is taken, the stalk which is going to be planted upon is called scion, and the plant which the taken plant, the scion is going to be made connected with. So that is called the stalk. So stalk will contain the root part, and the scion will contain the stem part. So the stalk part will be providing the nutrients, and the scion part will be providing the quality products. Okay, so this with that, after connecting it with the vascular bundles united, so you just have to tie it so tight with the plastic holdings or some cellophane tape or any duct tape or anything like that. So that makes sure or enables the sticking of the structure, the scion and the stalk together. Okay, so this is other vegetative propagation, artificial in nutshell. So next thing that you're going to talk about is tissue culture. Tissue culture is a technique which is implemented on limited basis. Why in a limited basis? It involves a lot of laboratory procedures. So what is it? What are the laboratory procedures involved in it, and why is it in here? We are going to form a new plant from the plant's vegetative parts, but they are in the near extinction or it is already extincted. So we just need a few living part of the tissues, okay, living part tissues of the plant. So these tissues are denatured, okay, denatured. It is harvested from the plants, or if it is going to be the only plant which is available, we'll harvest them from the plant, or it is already harvested that the plant is no more. We use that plant part and then surface sterilize them. Why? We have to get rid of any microbes so that it will not cause any damage to the tissue which is going to form. So we add it to the medium, okay? We add it to the medium, which will cause the process of de-differentiation. Okay, we need to understand three things over here. Okay, we need to understand differentiation. Okay, another one is de-differentiation and re-differentiation. I hope you all understand what I'm trying to say, because okay, I'm writing them down for you so that you will not get confused. So differentiation is a process where you study the 9th standard under the chapter tissues, the meristematic tissues, which constantly undergoes multiplication and then increase in number. They will acquire, they will actually perform one particular function. They will take up a function and they become that particular cell. So that's where we saw parenchyma, chlorenchyma, sclerenchyma tissues and arenchyma tissues. So those are different, those have changed from the meristematic tissues to those simple permanent tissues only by the process of differentiation. Now, what if we could reverse the process of differentiation? So this differentiation, when it is reversed and from the simple permanent tissue, if it is converted to the meristematic function, then that is called de-differentiation. So we have certain chemicals which could reverse the process of differentiation procedure that the plant part have already undergone, and then make it a undifferentiated mass of cell. Okay, so that is called callus. So that is called callus. I repeat, C-A-L-L-U-S, callus. So after achieving this part, we just have to direct them. So it's just a pot is already made, and from that pot we destroy the pot, and then we make soil particles, adding water to it, and then we'll add some more nutrients to it, and then we'll make it a new pot, like a different shape. Okay? So that is tissue culture. So over here, what happens is, after the de-differentiation of callus formation, which will have tissue available in the nutritive medium, it will be stimulated to undergo differentiation again. So this, okay, to differentiate now, it has already got differentiated. Now we have de-differentiated it, right? So it is not called differentiation again. So it is undergoing differentiation again. So it is called re-differentiation. So that's why we use the word re-differentiation. So this process is executed, and from the callus with proper nutrient provided to the callus and then a stimulation. So they will develop plantlets. So plantlets are smaller plant structures. So these plant structures are harvested or taken out and conditioned to by keeping them in the pot, and then make them sustainable for the external atmosphere, because these are not from the seeds after going through the hardship of life and then like coming to the pot. No, this is taken from the laboratory. So we need to swoop in, like, ease them into the society under environmental culture and stuff. So this process is called hardening. Okay, so this is the process of tissue culture. So why is this done? The tissue culture process is done for main reason where the plant or an organism, or especially the plants, so when they are in the process of extinction, okay, when you want to rethaw their existence, you mean like, revive their existence, you just need a small sample of living tissue of that plant with which we can recover the whole plant all over again. So this is found to be one of the most interesting and a very beautiful thing that you can possibly do with the technical things that you can learn. Okay, so this is about tissue culture. So the next thing that we are going to talk about is sexual reproduction. So the previous part, we have clearly explored about asexual reproduction. The next thing we are going to talk about is sexual reproduction. So we have already discussed about sexual reproduction in detail. Okay, the introduction about in detail in the previous videos. So if you have not checked our videos, so please check the previous video regarding this why do organisms reproduce chapter. Okay, so now when we talk about sexual reproduction involves two individuals. That's what we have already determined. Okay, so these two individuals are going to produce gametes, and these gametes are going to unite. And then these united gametes will give rise to a new individual. So thus forming a new individual. So such individual will be having more possibilities of variation, survival probability. So that's why sexual reproduction is one of the most important aspects when it comes to evolution. So this is one important point which you should remember for the next chapter. Okay, that is hereditary evolution chapter. So when you talk about sexual reproduction in organisms, it happens in both animals and plants. So we will see about sexual reproduction in plants needed, and then we'll go to the animals, which include human beings. Okay, sexual reproduction in plants actually involve two organisms. That's what I told you, two organisms. So two sex cells are required. One is the male reproductive sex cell, and another one is the female reproductive sex cell. So the male gamete here will be, okay, male gamete will be produced from the pollen grain, and the female gamete will be produced from the ovule. Okay, that will be taken care. Okay, they'll be united by a process called pollination. So the male gamete, the male and the female gamete, okay, are transferred, and that process called pollination. After that, the fusion of the male and the female gamete is called fertilization. So the fertilization happens. So from the fertilization happens, they will form seed, and the seed will be enclosed by the fruit. Okay, so that part is called the ovary. So ovary becomes the fruit. So now we'll see the parts in detail. Okay, shall we? So the ornamental flower actually has a peduncle or a stalk. Okay, and then this is the sepals. Okay, or the corolla, calyx, we call it calyx, C-A-L-Y-X, calyx. And this here is the petals or the sepals. Or sorry, petals are corolla, C-O-R-O-L-L-A, corolla, calyx and corolla. So now, if you see, the male structure actually involve particular things, and this is the female reproductive structures. Okay, so we will see them in detail in the next slide. Okay, so now this here is the male reproductive structure. So if you see all together, it is called stamen, and the stamen includes anther and filament. So anther will have special tissue that will be producing the pollen grain. So that's the pollen grain. So this pollen grain will contain three cells. One is the tube cell, and there will be two male gametes. I repeat, one tube cell, another two will be male gametes. So the tube cell will be the whole pollen grain, literally, and the male gametes will be present in the tube cell. Okay, and this blue color structure is the tube nucleus. Okay, I hope you people can see. See, this here is the male gamete, and the blue color structure over here is the tube nucleus. Okay. Now, we'll see the female reproductive structures. The female reproductive structure involve the carpel. That's the female reproductive structure. The carpel involve stigma. That's where the sticky substance and the nutritive material is actually available, and that has a specific function. We'll see them when they have the interaction part, okay? And then that extends to form a style, and that ends up in the ovary. So this ovary is going to develop into the fruit, and the inner structure you see something colorful, okay, like a rice grain. So that's the ovule, and ovule actually consists of a female gamete, and these are the polar nuclei, okay, and this is called filiform apparatus. So these structures actually constitutes the female reproductive part. Okay. Now, what happens? What happens is pollination. Okay, we'll study about pollination in the upcoming slides and different types and everything. But I'll tell you what is pollination in brief. So, the movement of the pollen grain from the anther and then gets deposited on the stigma is called pollination. And in this process, okay, the pollen grain will develop a tube-like structure, So this tube-like structure, okay, will be formed from the tube cell, and that will help in the descending of the male gamete, and then so that it will help them to reach into the ovule. So in that, when the tube nucleus literally follows or leads the pollen tube, and the male gamete which is found over here, okay, will be able to reach the ovule. So one of the male gamete fuses with the female gamete and then forms the embryo. That's the embryo. And the second male gamete fuses with the polar nuclei and then forms the endosperm. So the embryo and the endosperm constitutes together to form the seed, and the integument forms the seed coat. Okay, that encloses to form the seed. Now the ovary will be forming the fruit. So this is the sexual reproduction in plants. Now, when we go a little further, okay, this pollination is one of the important things. I'll finish pollination, then we'll go to the remaining slides. So the pollination is one of the most important things. Why? Because the transfer of pollen grain from the anther to the stigma, if that happens in the same flower in the plant, okay, so then it is going to be self-pollination, where it has reproduced a help reproduction by itself. If it happens with another flower from another plant, then it is called cross-pollination. Okay, so when the pollen grain reaches the stigma of the flower of the same plant, it is called self-pollination. If it is a pollen grain from one flower of one plant and then another flower of another plant, this is called cross-pollination. These are two different types of pollination, and pollination is usually helped by several agents like wind. Okay, we call them wind pollination, and water pollination, insect pollination, and bird pollination. Okay, so these are the types of pollination that we have learned. Okay, so now when we go to the next topic, which is the male reproductive unit. So male reproductive unit involves human being. Okay, we are going to study about human being. Human being, like hooray, because why? Because we are one of the complex organisms, and we get a chance to study about ourselves, and we are going to learn something in brief about male reproductive system, and the detailed part will be like followed up in the next set of videos. Okay, so the male reproductive system includes penis, which is the muscular organ with no bone in it. Okay, no bone assisted in human being. So that is going to be the copulatory organ. What is copulation? Copulation is a process where the union of the male and the female for the deposition of the gametes into the female reproductive system. So that's the muscular organ penis. The scrotum is a muscular bag of skin that holds the testis in place. See, I'll give you one important tip about this. The scrotum sac is actually seen outside in the male. Why? The testis is also found outside the body. Why? Because the testis requires less than 1 to 3 degrees centigrade of that of a body temperature to perform the gamete synthesis. So that process requires them to be get detached or kept away from the body so that it can be kept cool. So that's why it is the scrotal pouch that keeps them alive, outside. Okay, keep them alive outside, literally. And then the testis is the testicular tissue that holds the seminiferous tubules, and these seminiferous tubules actually contains the reproductive tissue, the cells. And then comes the epididymis, and then vas deferens, and then prostrate, and then seminal vesicles. So these are some of the structures which is going to support the process of male reproduction. And when it comes to the reproductive system, the muscular penis and the urethra, okay, both of them are part of two systems. One is urinary or excretory system and the reproductive system. So why is that so? It is because that it acts as a common passage where the excretory material is also taken out through the same passage, and the epididymis also comes out into through the same passage. That's why it is called a urinogenital. So this is male reproductive system in nutshell. So we will see all of them in the about male in detail in the next video. Okay. So the next thing we are going to talk about is female reproductive units. Female reproductive units is one of the most complex. So why is it complex? The male's process, the male's responsibility is just produce male gamete and then deliver them. Okay, but the female is where receive it and conceive and let them develop in their body themselves. Okay, so that includes several structures. One include vagina. So vagina is a passage. So vagina is an opening, and that leads to a passage. Okay, vaginal tract, and that tract will lead to the peduncle cleft. Okay. So this structure, this structure, cervical structure, will literally lead to the uterus. So the uterus will be like, will be the supporting structure where the developing embryo into the child is going to happen. Okay, and these structures will be like, protected from the outside in the vagina by labia major, labia minor, vulva, and clitoris. So these are the these are mostly nothing but the modified form of male reproductive structure. So when you talk about labia major and labia minor and clitoris, these are just secondary modification or modification of male reproductive structure in female because of the sex hormones found in the female. Okay. Now, Bartholin gland helps in the easy copulation process, so that the copulation will be lubricated. So that's the purpose of Bartholin's gland. So now we have seen the female reproductive unit in nutshell. Okay, so we will see the other structures in detail and their functions from the ovary. So this is the ovary part, and then this is the fimbriae part, and this is the fallopian tube. Okay, so all of them in detail in the next slides. Okay, in the upcoming things. So now, what is this asexual reproduction? Give an example. Okay, when you talk about asexual reproduction, I've already given you a lot of things. Like I've told you a lot of things about asexual reproduction. So we will see what is the answer. So organisms which chose to reproduce asexually by different means. Okay, let's say for an example kind of binary fission which you already saw. So when you talk about budding in hydra and then fragmentation in fragmentation in Spirogyra, and then planaria includes regeneration, and then ferns and fungus involves spore formation, and vegetative propagation includes onion, grass, and potato. All these things involve asexual reproduction. What are the advantages of tissue culture? So when you talk about advantages of tissue culture, it is, I've already told you again. So when the plant is in extension. Okay, excuse me a minute. So when the plant is in the grims of extinction, so we will try to revive the plant. We'll try to revive the plant. So when we revive the plant, that is one important thing that we can do. So what if we can produce, what we can have, we have a lot of other advantages. So one includes grow more plants in short time. So that's one great thing. Okay. So only a small amount of initial plant tissue is required, which means like you don't need a mass amount of tissue to produce a single plant. So this plant will, even if it has died because of virus, this plant which is going to be formed from the ex-plant or the vegetative part will be devoid of virus or any kind of disease. It will be disease free. Okay, and it doesn't. So even if you see when plants are supposed to be grown, they are grown only on particular seasons. But when it comes to this kind of plant tissue culture, it is not required that the plant should be grown only at a particular time period. So it can be grown at any time. So that really makes the thing more beautiful. I repeat. Okay. So these are the advantages of plant tissue culture. So I'm really happy to have you people with me. I hope you have learned something. So if you have any questions, I advise you personally to post them in the forum. We have an Ask IITians. We have dedicated teachers who can actually answer your questions at any time. And even your friends who also can post questions, and you can direct them to a particular faculty also. So keep looking for our videos, and then happy learning. Thank you so much.